Let's rock and roll. How's everybody doing? We got a massive class today. This is what I'm talking about. I like 40%. All right, so exams, the first part of them are graded. I didn't get a chance to do the second part, so I'll have that definitely for you all on Thursday. Okay? Who thinks you failed? Because you all did. just being honest. Okay. So on the what's today? Today's Tuesday? It is not well. Apparently, at my daughter's daycare, like McFeet, they can't say Valentine's Day. They have to say it's Friendship Day. It's Friendship Day. I'm like, let's promote love. Like, of all things, like let's go ahead and promote love, because this seems like something that's worth encouraging. But that's just me. How's it going, David? Okay, so sometime last week, probably about a week ago, we started talking about the big ideas, right? So we have gotten through kind of our intro, introducing chemical reactions, talking about the classes of enzymes, and diving into some reactions that we're going to see over and over this semester, like these themes. We've now moved on to thinking about metabolism, right? And we started introducing kind of like the background and some of the important concepts that we're going to see recurring in metabolism. And it really boiled down to 5 main principles. And we got through comfortably, we got through 3 of them, and we're going to finish the last 2 today. The first one is that molecules are degraded or synthesized stepwise, right? And these are our metabolic pathways. We didn't dive too deep into this because we're going to spend a lot of the semester talking about this, but 2 points pop out to me that I think are really, really worth knowing. One is that honestly, it allows for evolution to be clever, right? So, for example, we're going to see today in glycolysis that if we didn't go through this stepwise mechanism to convert glucose to pyruvate, we wouldn't be able to have the opportunity to create ATP along the way, right? So having these steps allows evolution to create reactions that are really beneficial. Okay, and that's what I mean by chemically clever. And also creates opportunities for branching pathways. And this is, if you remember on Tuesday, we saw that big map, right? And all of these pathways branched off of other pathways, right? It's an interconnecting network of reactions. So having all of these steps along the way allows interaction between these pathways, right? So it allows us to eat fats or sugars or proteins, right? And use all of them, and from each of them, be able to basically do anything else, with fats being one main exception, okay? So it creates opportunities to be able to create other molecules, basically, okay? The second big point was that ATP is the central energy currency of all living organisms, okay? And there were 3 important points that popped out here. One is that it creates a uniform template that enzymes can use to recognize one central molecule, right? So instead of having like 15 different molecules that all are used by enzymes to provide energy for chemical reactions, in which case enzymes have to be able to recognize one or more of those. In this case, since every single enzyme basically uses the same template, then we can have this scaffold that's generated that's always able to recognize that single central molecule. And you may remember from 523, I suspect Dr. Hurlbert mentioned the Rossmann fold. It's this very, very, very well conserved structural motif that when you see it, you can be quite sure you're looking for an ATP, or at least an ADP, functional group, okay? There's this idea we introduced of the phosphoryl transfer potential, which really simply boils down to the free energy of hydrolysis of a phosphate group, right? And we notice that ATP is centrally positioned, which means that there are molecules we make in the cell that are able to create ATP from ADP. We're going to see 2 examples of those today: phosphoenol pyruvate and 1,3-bisphosphoglycerate. These are 2 molecules that gets made during glycolysis. And since they're made during glycolysis, we'll take advantage of them to then create ATP. But in most cases, ATP has a higher phosphoryl transfer potential, which means it's able to then take its phosphate group and give it to something else in a spontaneous manner. So ATP can be made in this substrate level phosphorylation by molecules that have a higher phosphoryl transfer potential than ATP does. But once it's made, it can then power lots of reactions and give its phosphate to lots of other things. And another really important one we focused on is the idea that it's thermodynamically unstable, meaning hydrolysis provides energy. It's a spontaneous reaction that generates energy, but it's kinetically stable. And this is important because if it was spontaneous and kinetically unstable, then that would mean that it would immediately react and we can never store the energy, right? So we make ATP and it immediately gets hydrolyzed. That's a problem, but it's kinetically stable, meaning despite the fact that there's a spontaneous reaction, the activation energy is so high that kinetically it's very slow. And it requires an enzyme to decrease that activation barrier and make the reaction faster. So this is an important theme that we're going to see not just in these phosphate compounds, but really in cofactors in general or molecules in general that are like functional group like they're able to carry functional groups. Okay, we'll see a couple more examples today. The third one we talked about here is that ATP can be formed by the oxidation of carbon-based fuels. Okay, we didn't spend a ton of time thinking about this, but what I do want you to remember, and we did talk about, is this idea that the more reduced carbon is, the more energy that can be produced during oxidation, right? So methane with 4 hydrogens surrounding carbon is the most reduced form of carbon. And carbon dioxide, where it's fully oxidized down to basically deficient 4 electrons, there's 0 energy from combustion, right? So the more reduced it is, the more energy potential there is for oxidation. This is relevant because at the end of the day, our central pathways, our catabolic pathways, they produce energy, they produce ATP by oxidizing carbon in these carbon-containing molecules. Primarily, we're going to think of mostly glucose or fatty acids. Fatty acids are much more reduced than sugars, right? Fatty acids have very reduced carbon relative to sugars, right? Sugars are decorated with lots of alcohols. So our fatty acids are the ones that are going to produce a lot more energy, and therefore they're better at storing energy than sugars. Okay, and here is an image that we saw last week that kind of showed the positioning of ATP relative to other compounds in this phosphoryl transfer potential idea, reinforcing the idea that it's centrally positioned. Okay, now moving on to the fourth point. And this is the idea that although many reactions occur inside of a cell, there are themes that make our life a lot easier. Okay. So we did touch on this briefly at the end of the day on last week. But so one of these themes we've really kind of already seen, and that's that there are a set of cofactors that do the same thing for lots and lots of reactions, right? And these tend to be the vitamins that we have to eat, right? These are molecules that our body is not necessarily good at making, so we get in our diet, okay? And we've talked about most of these before. TPP, it's really good at doing aldehyde transfers. Flavin and nicotinamide, right? So our FAD and our NAD cofactors. These are really, really good in carrying electrons in our oxidation reduction reactions. Pyridoxal phosphate, incredibly important for transferring amino groups. That's our PLP. Coenzyme A we touched on. We're going to see that this is incredibly important for moving around carbon-containing groups. We haven't talked about biotin. We will. We briefly talked about tetrahydrofolate, folic acid. This is really important for moving methyl groups in different oxidation states. And then vitamin B12 you saw in one of the problem set questions, and it focused, so it basically allows us to rearrange carbon skeletons, right? These are central themes that we see over and over. So if you understand how it works with one reaction, you understand how it works with all of them. In addition, we have these recurring activated carriers. Okay, so some of them are really obvious we talked about, right? So activated carriers, ATP is our activated phosphoryl carrier, right? It carries a phosphate group. NADH, FADH2, FMNH, right? These are all the nicotinamide and the flavin containing cofactors. These are electron carriers. Coenzyme A and lipamide are both carbon acyl group carriers, right? So we won't go through all of them, but the idea is that the same theme recurs. If you ever need to transfer, let's say, an acetyl group from one molecule to another, it's really likely that either coenzyme A or lipoic acid are going to be involved. If you need to transfer a phosphate from one place to another, from one molecule to another, it's really likely that ATP will be involved, right? Because life has evolved to use these recurring carriers, okay? They are kinetically stable, meaning that once we make these molecules, once we have phosphate on ATP, once we have electrons on NADH or FADH2, once we have an acetyl group on coenzyme A, it's kinetically stable. It's not going to spontaneously hydrolyze really quickly. However, hydrolysis is typically spontaneous. So thermodynamically unstable, kinetically stable. Okay, so here we're going to see, just I guess really briefly, just see some of these reactions that we've seen before, right? So we spent the better part of 2 weeks talking about some of these reactions. Here we're just going to summarize them. So in situations where you want to oxidize an alcohol to a ketone, that's an oxidation reaction, so we need to use one of our activated electron carriers. That could be either nicotinamide or flavin. Okay, it tends to be true that the nicotinamide, our NAD cofactor, tends to be more important in oxidizing oxygen-containing functional groups, whereas the flavin tends to be important in oxidation or reduction reactions that involve carbon-carbon skeletons. Okay, so some of these themes aren't as obvious, but once you see them, you can readily use them. Okay, once you understand the chemistry of FADH2 and NADH, then it's the same thing over and over. You don't have to relearn it, right? We know that for flavin, our reactive sites are the two nitrogens positioned here. We know that they're linked electronically through these pi bonds. So it always involves hydride transfer. So a hydride would attack at one of these two positions. The electrons would reposition themselves, and then this nitrogen will ultimately pick up a proton. Over here, we know that this is the reactive site. A hydride would come in, react here, electrons would move down to this nitrogen, and they'd be stored there, right? Once you understand how this chemistry works, you don't have to relearn it for every reaction. I briefly mentioned last week that R group on NADH we're showing there at the kind of bottom right corner. So this can either be a hydrogen if it's NADH, or it can be a phosphate if it's an NADPH, okay? And I point that out because that's another really common recurring theme, is that when this is a hydrogen, that's NADH, and this tends to be important in catabolism, in catabolic reactions, pathways that produce energy. But if this is a phosphate, it's NADPH, and this tends to be important solely in anabolic reactions, so when we're consuming energy, right? This is really relevant because it means that systems can evolve, life can evolve, so that opposing pathways are similar but divergent enough that they recognize different things. The presence of this phosphate group here would prevent catabolic reactions from occurring if this is in the reduced state, right? Because these enzymes won't look for this phosphate, they're going to look for the alcohol functional group. Okay, coenzyme A. Once we get to the end of glycolysis and we start thinking about transitioning from a glycolysis into the TCA cycle, we're going to start seeing coenzyme A present a lot, especially when we start dealing with fatty acids, because coenzyme A is the most critical acyl carrier, carbon chain carrier in our cells. right? So the idea is really simple. Is really simple. We have an ADP, right? And we've seen this before. Coenzyme A has an AMP or ADP functionality. NADH, FADH2, right? So many molecules contain this AMP because it's a recurring theme that enzymes can recognize, right? So it has this AMP so that we don't have to reinvent the wheel for enzymes to recognize something. Attached to this AMP is a phosphate and then a molecule called pantothenic acid, which is something that we get in our diet. Okay, and then attached to pantothenic acid is simply a 2-carbon unit containing a nitrogen connected through a peptide bond, and then it has a reactive sulfur at the end. Usually, we ignore everything in coenzyme A except this reactive sulfur, right? It's a reactive thiol. At the end of the day, coenzyme A boils down to this reactive thiol group. The rest of this is decoration for enzymes to recognize. And that's true for lots of our cofactors, right? The rest of NADH, the rest of FADH2 is really just decorations for enzymes to recognize and position molecules properly. This reactive thiol will readily react with a carboxylic acid or any other ester, right? And it can replace, so this sulfur can replace the oxygen in an ester or a carboxylic acid linkage to make an acyl-CoA. So here is acetyl-CoA. It is coenzyme A covalently linked to acetic acid by a condensation reaction. Okay, this is relevant because this is stable, right? To hydrolyze this, we have to go through a transition state that includes a negative oxyanion, right? So just like phosphate groups being kinetically stable because of the high activation energy, the same idea goes here. To hydrolyze this, we need to go through an unstable transition state. So the only way to hydrolyze it kinetically is to decrease the activation energy by putting it in the context of an enzyme active site that can stabilize that. But hydrolysis of this is spontaneous, okay, which means that once we produce this, it's stable, it can move around, it won't fall off. But once there's an enzyme around that can hydrolyze it, hydrolysis of this is spontaneous, meaning that once we want to get it off, we can get it off. And it'll actually produce energy when we do so. Okay, other ideas. So other things that we see conserved are the idea of protein structures, right? And we're not going to really get into this quite as much this semester as maybe y'all did last semester, but this is one incredibly common theme, and this is the Rossmann fold, right? So it's this beta, alpha, beta repeating pattern. And if you look here, you'll see it over and over. You have a beta sheet, an alpha helix, a beta sheet. You have a beta sheet, an alpha helix, a beta sheet. You have a beta, let's see, where is that? A beta sheet, an alpha helix, a beta sheet. This repeating pattern over and over. It's very, very recognizable. And this tends to recognize molecules that contain nucleotides, specifically dinucleotides. Okay, but it's really good at recognizing molecules that contain nucleotides. So if you see this structural motif, you have a really good idea for what the enzyme does, at least to some degree. Okay, this is the last common theme we'll talk about is just themes in the chemistry, right? So things really boil down to 6 types of reactions, the 6 types of enzymes, 6 classes of enzymes. Despite the fact that there are thousands of reactions, all of them categorized into 6 categories. So it simplifies things really quickly. And we start thinking about common functional groups and common reactions, right? So there are themes in nucleophiles. There are themes in electrophiles. We really don't find anything else. Nucleophiles, we have deprotonated alcohol groups. We have deprotonated thiol groups. We have carbanions. We have amines. We have imidazole. We have hydroxide. There's not much else. This is kind of it. Electrophiles, we have a carbonyl. We have a phosphate group. We have a shift base. We have a proton. There's not much else. Okay, so what I'm trying to get at is that if you recognize themes and patterns, it makes metabolism a lot easier. Finally, metabolic pathways are highly regulated. Okay, and where's it at? Okay, so this kind of boils down to three different main principles, right? It can be regulated at the level of transcription. Okay, and so here the amount of enzyme, the quantity of the enzyme can be controlled at the level of transcription. Okay, or at the level of post-transcription, right? But the idea is we can control the amount of an enzyme present. We can control the catalytic activity, so how active an enzyme is, using allosteric regulators, using chemical signals, right? At the end of the day, the two really kind of big things are hormones and then some signal about the energy status of the cell. Hormones, if you've taken other classes that talk about energy in a whole organism, maybe you've talked about this, right? So the idea of insulin and glucagon. Insulin is secreted by the pancreas under conditions where you have a high blood sugar level, right? And that tells your cells to bring that glucose into your blood or into the cells and activate pathways that use that sugar for energy. Okay, if you have low blood sugar, insulin's not secreted, glucagon's secreted, and that tells your cells to stop taking up glucose and instead mobilize glucose in the liver to send to your brain and other tissues that absolutely have to function, right? So hormones are really important signals. Inside the cell, things can respond to the energy status of the cell, which we'll look at here in a second. The idea here is that right, so like the relative amounts of ATP versus ADP and AMP is a really good signal of how much energy the cell has. And if it has plenty of energy, there's no point making more. Okay? We can prevent opposing reactions, wake up, by compartmentalization. We can so glucose can either be made or degraded depending upon the needs of the cell, right? And yes, we can activate certain enzymes if things are happening in the same compartment. But it's also easier if we just make sure that these opposing reactions are separated, right? So inside the cell, we're going to see that fatty acid synthesis happens in the cytoplasm, whereas fatty acid degradation to make energy happens in the matrix of the mitochondria. So those enzymes are physically separated and they can't compete with each other. And I really like this. So this is a cool diagram that I could spend probably the rest of class today talking about. It's this idea of metabolic flux, okay, and how the activity of certain pathways responds to the amount of stuff in a certain pool, right? And because I would talk about this forever, I'm not going to get going on it. But the general idea is think about this as a roadmap, right? And how much traffic is on a certain road, right? And if you're driving along and you're using Waze or Google Maps or whatever the hell you use, right? If there's congestion up ahead, you get rerouted down a different pathway, right? This is the idea of metabolic flux, is that if there's congestion in one place, pathways will alter such that metabolites shift to do something else, right? If there's plenty of energy, then things will shift to store energy. If we need energy, then the activity of pathways shift to make more energy, right? So everything responds to signals within the cell. And this idea of movement of energy, movement of molecules is metabolic flux. Okay? So this is all just reiterating what I just said, so I won't reread it again. But the one thing I want to really stress here is that this, what I have in red, is that homeostasis is not equilibrium. This is an important thing to remember, okay? Equilibrium for living systems is death. It is like if equilibrium is established, that means you're dead, right? There's always flux. There's always movement of molecules. Things are never at equilibrium. There's always at least one pathway pushing things forward, okay? But homeostasis means that things can reach relatively stable levels, right? But that does not mean equilibrium because things are still responsive, okay? Again, I'm not gonna really dive deep into right here because I've already kind of talked through that. But I do wanna finish up here with this idea of controlling catalytic activity through the energy status of the cell, okay? So I mentioned that this is the relative levels of ATP versus AMP and ADP. Okay, and there's really two ways of cataloging this. Okay, we call it the energy charge or the phosphorylation potential. And quite frankly, they mean the same thing. They just give a little bit different numbers. Energy charge is thinking about the total concentration of hydrolyzable phosphate versus the total concentration of adenosine. Okay, so in the denominator here, we're basically adding up all of the adenosine in the form of ATP, ADP, or AMP. On the top here, we're adding up the total amount of hydrolyzable phosphate. ATP has 2, ADP has 1, which is why there's a 1/2 in front of ADP. Okay, and it ends up being a value from 0 to 1. If the value is 1, all of the adenosine is in the form of ATP, and we have a massive energy overload. If it's 0, everything is in the form of AMP, and we desperately need energy, right? And you can see here that when you look at the activity, the flux through pathways that use ATP versus flux through pathways that produce ATP, you can see where they meet really gives you a really good sense of what the cell wants this energy charge to be, right? So you can tell here that the cell wants the energy charge to be around 0.85, 0.9, because at this point, this is where the pathways converge. This is where they're both equally active. So that's a point where it's likely that that's what the cell wants. If you have this much ATP around, things are functioning really, really well. All right, so with that, I think that, yep, so that finishes up this part of it. Y'all go ahead and take like 10 minutes to work on the activity from last week and finish it up if you want to, and then we're going to jump into glycolysis.